Hi guys, we are back with our today's questions. Today we will be talking about nitrogen fixation, one of the primary events in the entire nitrogen cycle which provides nitrogen to the entire biosphere. So the first question, it says that nitrogenase, right, the enzyme which fixes the atmospheric nitrogen, that is the nitrogenase coded by the NEET gene, it requires which of the following? So for fixation of nitrogen requires what? So it is A, oxygen radicals. We know that they are toxic, so it doesn't require oxygen radicals. Calcium as such does not have any direct role in the fixation of nitrogen. Energy as ATP. Yes, for the time being we know that for fixation of this N2 to NH3 we require about 16 molecules of ATP. So it requires energy in the form of ATP. Sulfur and manganese, again no direct role as such for nitrogenase. So nitrogenase requires energy in the form of ATP. So answer here is option C, energy as ATP. Second, it says which of the following is the cofactor for nitrate reductase. So we know that enzymes, they have cofactors. Sometimes they are organic, sometimes they are inorganic. Nitrogenase, we know that has a molybdenum based cofactor. So here you can see the option D, that is molybdenum. So the cofactor for nitrate reductase is your molybdenum, that is option D. Third, it says the leg hemoglobin. Right, now this leg hemoglobin we know that is required for binding with the oxygen, so that the availability of oxygen in the area where nitrogen is being fixed, that is reduced. So it sequesters the oxygen from there. So we know that it is sensitive. The enzyme nitrogenase is sensitive to oxygen. So leg hemoglobin is required for which of the process? So it requires for the process of nitrogen fixation, so that it can remove the oxygen from the place where this nitrogen is being fixed, so that the process it continues unhampered. So it is not required much in photosynthesis, in fatty acid synthesis or in respiration. So here the leg hemoglobin is required for A, nitrogen fixation. Fourth, it says nitrogen fixers are generally, right, here we are talking about biological nitrogen fixers. So nitrogen fixers generally we know that they are bacteria. So here the options which you have is your algae, no they are not, protists they are not, fungi they are not. So most of the nitrogen we know is fixed by the class that is eubacteria, right, the domain eubacteria. Most of the nitrogen fixers, you may talk about cyanobacteria, you may talk about say rhizobium, azotobacter, clostridium, pseudomonas, n number of organisms, bacteria which fix the nitrogen, it belongs to the class of eubacteria. So nitrogen fixers generally out of these four options they are eubacteria. Thank you and stay tuned with me, Professor Girish Kukreja, for more such questions.